Assalamu alaikum dear students. Welcome to the course Principle in Animal Life 1. This is lecture number 31. The topic is exploring levels of protein structure. Discussing agenda for the topic is primary structure of proteins and secondary structure of proteins. Lecture outcome. After watching and listening this lecture, students will know about the different levels of protein structure. Introduction. There are a total of 4 levels of protein structure. In this lecture, we will discuss the first 2 levels, and in the subsequent lecture, we will discuss the remaining 2 structural levels of proteins. In spite of their great diversity, proteins share 3 superimposed levels of structures known as primary, secondary, and tertiary. Proteins are made up of only 20 different types of amino acids, but they are the most diverse biological macromolecules. They have hundreds of thousands of different types on the basis of their shape, on the basis of their structure. So in this lecture we will analyze which type of chemical interactions are present in the structure of proteins that gives characteristic shape to the proteins, characteristic properties to the proteins, and enables them perform their function in the best of ways. Now, a 4th level, quaternary structure, is formed where 2 polypeptides or more than 2 polypeptides that may either be same or different combine, and it is the 4th level of protein structure. Be sure to study this figure thoroughly before going to the next section. So it's an important topic. First, the primary structure. Primary structure is a linear chain of amino acids. Amino acids are connected to each other with the help of the peptide bonds. Peptide bond is a covalent bond, and it takes place between the amino group of one amino acid and the carboxyl group of another amino acid. So the primary structure of a protein is its sequence of amino acids. As an example, let's consider trans thyroidine, a globular blood protein that transports vitamin A and one of the thyroid hormones. On the right side of your screen, you have an illustration of trans thyroidine protein. This is the primary structure of trans thyroidine protein, and you can see it's the sequence, a linear sequence of amino acids. Trans thyroidine is made up of 4 identical polypeptide chains, each composed of 127 amino acids. So here is one of these 4 chains. Each of the 127 positions along the chain is occupied by one of the 20 amino acids indicated here by its three-letter abbreviation. So these are different types of amino acids linked together with the help of peptide bonds, and it makes up the primary structure of protein. The primary structure is like the order of letters in a very long word, just like this is a word where the letters are present. Similarly, the primary structure is a linear structure where the amino acids are repeated again and again. If left to chance, there would be 20 raised to the power 127 different ways of making a polypeptide chain, 127 amino acids long. So this basically means that how many different proteins can this chain make if you remove and rearrange this order, then it can make up 20 raised to the power 127 different types of polypeptides. Just the rearrangement of those 20 amino acids, it gives you a very good idea about the diversity of proteins. Although they are made up of those 20 types of amino acids, but it is the arrangement, it is the sequence of those amino acids which makes them different from one another. And of course, the sequence is decided by the sequence of nucleotides in DNA, because DNA stores message for the synthesis of the polypeptides, and a single gene can prepare more than one type of polypeptides, courtesy of alternative splicing, where the exons change their positions. So a single gene can make up more than one type of polypeptides, courtesy of alternative splicing. However, the precise primary structure of a protein is determined not by the random linking of amino acids, but by inherited genetic information. So although this polypeptide has this enormous ability to become different because of the rearrangement of amino acids, this polypeptide made up of 20 amino acids, 127 amino acid long, where the amino acids can be rearranged and 20 raised to the power 127 combinations are possible, but it doesn't happen every time, because the genes, the sequence of nucleotides in the DNA determines the sequence of amino acids in proteins. Therefore, this sequence is predetermined and it is fixed and it is not haphazard. The amino acids doesn't arrange by chance in such a way The instructions for this precise arrangement is stored in DNA. The primary structure in turn dictates secondary structure and tertiary structure. Now the primary structure, it is the first level, and thereafter in secondary structure the skeleton of the amino acids they interact with the skeleton of other amino acids. So this precise arrangement helps to predict the secondary and tertiary structures of the proteins, because the amino acids facing each other and in close proximity will establish association with one another courtesy of their amino acid skeletons. Due to the chemical nature of the backbone and the side chains, the amino acids along the polypeptide, they interact with each other. In the tertiary structure, we will study 5 types of interactions which involves the side chains, and it is predetermined that where the side chain of a particular amino acid will establish an association, and therefore a mutation which only replaces one amino acid have drastic effects on the three-dimensional structure of the protein. So now we study the secondary structure. The primary structure is a linear sequence of amino acids, but this linear sequence has a role, or it determines the type of interaction in secondary and tertiary structures. So let's have a look at the primary secondary structure of proteins. Now in the primary structure of proteins, there is only peptide bond. Only peptide bond is there in the primary structure, and that peptide bond is established between the amino group and the carboxyl group of two amino acids. Here another type of interaction establishes between the skeletons of two amino acids. Remember that in secondary structure the skeleton of the amino acids is involved. The R group or the functional group is not involved in the secondary structure. So regions stabilized by hydrogen bonds between atoms of the polypeptide backbone. Now we have two types of secondary structures. One is alpha helix and the other is beta pleated sheets. In the alpha helix, the polypeptide is twisted and coiled like a DNA molecule, and there is interaction between the side chains of two amino acids lying in close proximity along the length of the helix. Now these coils and folds, collectively referred to as secondary structures, are the result of hydrogen bonds between the repeating constituents of the polypeptide backbone. Once again, remind you that in the secondary structure the amino acid backbone is involved and the side chains are not involved. Within the backbone, the oxygen atoms have a partial negative charge, and the hydrogen atoms attached to the nitrogen have a partial positive charge. Therefore, hydrogen bonds can form between these atoms. So here the bonding is between the oxygen of the carboxyl group and the hydrogen of the amino group, but this bonding is different from the peptide bonding, in which the bond forms between adjacent amino acids. Here the bond is formed between the first and every fourth amino acid in the helix. Individually, these hydrogen bonds are weaker, but because they are repeated many times over a relatively long region of the polypeptide chain, so they can support a particular shape for that part of the protein. Hydrogen bond is a weak bond, but there are so many hydrogen bonds, so their cumulative effect provides strength to the protein. One such secondary structure is the alpha helix, a delicate coil held together by hydrogen bonding between every fourth amino acid. So suppose one amino acid is present here, so it will establish a hydrogen bond with every fourth amino acid. Although each trans-thyroidine polypeptide has only one alpha helix region, other globular proteins have multiple stretches of alpha helix separated by non-helical regions. So a single polypeptide may both have helical regions and non-helical regions. Now, beta-pleated sheets. Some fibrous proteins such as alpha-keratin, the structural protein of hair, have the alpha helix formation over most of their length. The other secondary structure is the beta-pleated sheets. Now, in beta-pleated sheets, two or more segments of the polypeptide chain lying side by side. Remember, this is the beta-pleated sheets, and the segments of the polypeptide, they are lying side by side instead of the helix. And now this hydrogen bond will be established between them. Beta-pleated sheets make up the core of many globular proteins. Core means the inner region of many globular proteins, and dominate some fibrous proteins, including the silk proteins of a spider's web. This is the end of the lecture. I hope you would have gained some knowledge from this video. Your questions are welcomed not only on KCMS portal but also on WhatsApp groups. Thank you.